Hello, everyone. Thank you very much for the invitation. It is a pleasure to be here today. My name is Alex Bliu, and I am a senior biostatistician here at Health Canada. As a disclosure, the views presented here are mine and do not necessarily reflect those of Health Canada. This is the agenda for my talk today, and will focus on statistical approaches in pediatric studies, which aim to find the right balance between the visibility of conducting studies in children and the information required to establish treatment benefits in pediatrics. We need to minimize the number of subjects enrolled in pediatric clinical trials and the need to maximize the usefulness of the data obtained, while ensuring that the trials are visible and the results are robust and interpretable. As we know, running clinical studies in children is complicated given a number of challenges. But on the other hand, we would like to keep the level of scientific evidence at the same higher level as studies in adults. And in order to do that, we need sufficient data or large enough sample size with pediatric trials. But how do we come around that given the low enrollment rate of pediatric trials and the associated challenges with running these trials? Well, that is where the concept of extrapolation comes in which one tries to leverage available data from adults or older age groups and draw conclusion for the pediatric population, thus reducing the need to generate additional information to reach conclusion for the pediatric population. As with many other approaches, extrapolation comes with assumptions attached to it, in which we need to ensure some level of similarity between the source population, the adults, for example, and the target population, the children. There are different ways on how to establish such similarity and also different requirements depending on the extent of the similarity required. However, I will not focus on methods around similarity in this talk. The use of extrapolation ensures that we generate just the right amount of the data, and we use statistical methods to draw conclusion from the data. In terms of statistical approaches to use, well, one can use either a frequentist approach or a Bayesian one, and I'm not here to promote one over the other, and perhaps that would be a topic for another talk. And for the most part, the method to be used depends on the type of the extrapolation approach. Now, both frequentist and Bayesian methods serve the same purpose, which is to maximize the use of the data and make robust estimation of the treatment benefit in children. However, Bayesian approaches and frequentist approaches, they look at the issue from a different angle. Having said that, in this presentation, I'll focus mostly on methods that are unique to studies in children and extrapolation in particular, and not on the more sort of traditional methods used in clinical trials. So narrowing down to the statistical methods for pediatric studies, there is a wide range of methods available for use. Some of these are more traditional than the others, and some are new, and certainly there is lots of debate to which method is more suitable, but above all, it really depends on the study at hand. For example, Bayesian methods have gained popularity lately, not only for pediatric trials, but also for adult trials, and some of these fancy methods are even being used in developing COVID-19 therapies and vaccines. Type 1 error is another hot topic since Certainly for us as regulators at least, we don't want to wrongly conclude that treatment is beneficial, especially in vulnerable populations, such as pediatrics and rare diseases. We hear a lot about real-world evidence, and rightly so, since there is lots of data out there from observation studies, registries, and other sources. But how useful is that data in order to make causal inference? And we'll touch base on that as well during this talk. With that, let's talk a bit more about some of these methods, and let's start with modeling and simulation. So for those who know that modeling and simulation is more of a Bayesian method, they would ask, why is he talking about modeling and simulation before explaining what Bayesian is? And rightly so, but I thought I'd start where extrapolation first began, which is with PK and PK-PD studies. On that note, modeling and simulation is a powerful tool that can be used to examine study design aspects, derive dosing recommendations, and perform sensitivity analyses. Techniques for modeling simulations, such as PK and PD, clinical trials, and disease states have advanced significantly. Advanced approaches such as physiological-based PK or PBPK modelings are used to predict and select pediatric dosing regimens for use in clinical trials. In addition, for example, POP-PK and spare sampling approaches can provide valuable information on how drug clearance is influenced by factors such as age, gender, body weight, etc., to inform product labeling and use. And now with that, and now the new kid in the block, the Bayesian statistician. Well, the kid is not new, just not that popular yet, but it is getting there slowly but surely. Conventional frequentist methods require a relatively large number of subjects, with each product requiring its own sequential and often costly clinical trials. The Bayesian statistical paradigm is natural with extrapolation, as it permits the combining, or otherwise known as borrowing of the information, across different sources, such as the adult and the existing pediatric data. So say we are interested in estimating a treatment effect theta in a pediatric population. With a frequentist approach, one would maximize the likelihood of this treatment parameter theta given the collected data. Rather than simply maximizing at least this likelihood function to find a point estimate for theta, Bayesian methods instead add a prior distribution, summarizing what is known about the treatment parameter before data collection. A Bayesian then computes the posterior distribution parameter theta given the data via Bayesian rules using either probability calculus or, where closed form calculations are not possible, a numerical approach such as Markov chain Monte Carlo simulations are used. For those of you that have worked with statisticians, know that there is always assumptions to be made with any statistical method, and even with a simple linear regression, there are 4 or 5 assumptions that one must check, although I'm not sure if people check these assumptions anymore. This is even more so with Bayesian methods. There is a number of assumptions and specifications that one must make, and each of these assumptions should be carefully considered. Some of these include, for example, am I using the right prior? Would a better prior explain the known data? How much can I borrow? Where is the model borrowing from, and whether should I be borrowing at all? These are very important questions that should be properly addressed and validated when using Bayesian methods. And in this slide, we move to an application of Bayesian method with pediatric trials, the hierarchical model, also known as a mixed effect modeling in frequentist world. The idea around this approach is on how to incorporate information from different related studies, which fits very well with the concept of extrapolations, since after all, extrapolation relies on leveraging known information. A typical hierarchical model, for example, may include two levels, a patient level and an age cohort level. In a two-level structure, cohorts have different but related treatment effects or means. The cohort treatment effects are related through a common distribution. This relationship is at the heart of what is meant by exchangeable cohorts. In fact, the assumption of exchangeability means that cohorts have different but related treatment effects or means, and are then related through a common distribution. As you can see, there are assumptions on top of more assumptions with Bayesian methods. Hence, one needs to exercise extra caution with these methods and their applications. So let us now move to the type or source of information that we can use for pediatric studies and extrapolation in particular. When studies are required in order to generate efficacy data, one of the most important design features and decisions will be the choice of the control arm. This is usually expected to be either a randomized concurrent control, an external control, or even an uncontrolled study, perhaps. The choice will be influenced by the scientific questions identified in the study and in the extrapolation concept in particular. Whether we use external or historical controls with frequentist approaches, for example, to establish overall response rates or non-inferiority margins, or we use these controls with Bayesian approaches to construct, let's say, priors or establish thresholds, one should pay careful attention to the choice of these controls and how these controls best match your study at hand. Moving on to this slide where we have what regulators talk about a lot, the type 1 error. Are the results statistically significant? Is this a chance finding? In order to address these concerns, one should design usually a large trial, which, as we just discussed, is not feasible for many pediatric trials. So what can we do? What do we do? As always, it is all about finding a compromise. Can we lower the bar when it comes to the type 1 error rate? And instead of looking at the traditional 5% rate, can we look at a more relaxed rate, since that would indeed decrease the sample size and help on the feasibility of running the trial in pediatrics? Well, that depends on the type of the study at hand in general. But it turns out that with extrapolation, we can decrease the type 1 error rate to, let's say, 10% instead of 5%, because the likelihood of wrongly concluding a treatment benefit is less likely with extrapolation, because the treatment benefit has already been established in the adult population, let's say. What else can we do? What other solutions do we have at hand? Well, one could conduct small or underpowered studies and then combine the results using a meta-analysis approach. Alternatively, as we just discussed, one could use Bayesian approaches. In general, it is always more informative to look at the actual p-value rather than a binary form of significant versus non-significant. A p-value, for example, of 0.0 is much better than a p-value of 0.28, let's say. In addition, we could also use the totality of evidence to look at other measures, such as the effect size, confidence intervals, secondary endpoints, etc. And above all, to remember is that the p-value is not only the only matrix to look at. The benefit-risk assessment plays a very important role in decision-making. And here we go and move to another topic, the real-world data, real-world evidence. These terminologies have been very popular lately. These are powerful tools to make use of already existing data, and especially with pediatric trials and rare diseases, where it is very challenging to conduct clinical studies. There is no doubt that the evidence based on real-world data is very useful. But I think there is still work to be done as far as the regulatory claims go. There is still lots of work to be done to minimize any potential for bias and confounding with real-world data. At the end, the aim is to establish a causal link between the treatment and the outcome. And there is still work to be done with respect to the quality of the data and the statistical methods to analyze this data so we can reach a robust and reliable conclusion with the real world data and generating real world evidence based on this data. In closing, early and careful planning is important with any clinical trial in general, and even more so when designing a small clinical trial in pediatrics and when applying the concept of extrapolation, which tries to predict by projecting past experiences or known data. In addition to what type of pediatric data we need to generate and how we generate the data, we also need to pay attention at the quality of the data, of the adult data, and how to use the adult data for the purpose of the extrapolation. There is no doubt that early interaction with regulators will improve the efficiency and clarity of the drug development process and ultimately promote and enhance product quality. There is a need for harmonization of methods and strategies into overall drug development plan in order to improve the speed of access to new drugs for pediatric patients, and there is work being done at the ICH level, for example, ICH E11. Luckily, statisticians are here to help, and there are a number of novel statistical methods that we can use. I can draw your attention to the ICH E9 and ICH E9R1 addendum on estimates and sensitivity analysis. And with that, I thank you very much and for your attention, and I leave you with a beautiful fall picture of Gatineau Park here in Quebec, Canada. Thank you very much. Merci beaucoup.